Hello everyone, and welcome to this IFST webinar on nutrient bioavailability, processing the possibilities. I'm going to be your chair today. My name is Kate Halliwell. I'm chair of the IFST's Food Science and Nutrition Specialist Interest Group. We're the people who've put on this event today. I'm also the Food and Drink Federation's Chief Scientific Officer, which means that I overlook our policies in the food safety, food law and nutrition space. And I must confess that until quite recently, I probably hadn't thought much about structures when it came to nutrient content. I have a nutrition background, so very aware of the nutrient levels in foods, but probably outside of fibre, hadn't given structures much thought into the bioavailability. But it's something that I think is becoming increasingly obvious that it is something we should be thinking about, actually. So I am really pleased that we have got a great set of speakers today who are going to take us through at least the start of the conversation. So we will be hearing from Mike Adams, who is Head of Product Innovation at Camden BRI Group, Professor Paul Sharp, who is Professor of Nutritional Science and Head of the Nutrition and Dietetics Department at King's College London, and Eduardo Capuano, who is Associate Professor of Food Quality and Design at Wageningen University in the Netherlands. So you will be able to see full bios for our speakers if you want to find out more about them on the IFST website. So just before I hand over to our first speaker, a couple of bits of basically IT housekeeping. So this webinar is being recorded and it will be uploaded onto the IFST webinar hub after the event. Each of our speakers is going to talk for around about 10 minutes, but during that time, if you have any comments or questions that you want to ask, you can type those in the Q&A using the Q&A button. You'll find that at the bottom of your screen. If you like somebody else's question, you can also uptick that so that then I will know which questions that most people think are really interesting and want an answer to, and we can prioritise asking those. So after all of the presentations, we're going to run straight through them. So we'll then go on to the Q&A session. So we should have probably just under half an hour, I would imagine, for Q&A. So please do drop those questions in as you think of them. And then without further ado, I'm going to hand over to our first speaker, who is Mike Adams.
Thanks, Kate. Hopefully you can see my slide and hear me speak. I'm going to talk about a general overview of food structure and nutrient availability from a product developer's perspective. We've got some great academic speakers coming after me who are going to talk in more detail about their work, but I'm going to provide a kind of top-line background about the importance of structure and nutrient availability and some of the factors that as us product developers have an influence over. So why is structure important? Well, structure impacts food and beverages in a myriad of different ways. As you can see from this graphic, and this is just a selection of 5 of the things that structure has an impact on. We're talking about nutrition today, but obviously structures, whether natural or designed or engineered structures, have a huge impact on every aspect of food and beverage. And from a product developer's perspective, a lot of these are at the consumer. So consumers' understanding of the appearance, the mouthfeel, the flavour, the texture of product, all are significantly impacted by the structure. As I say, some of these are designed structures, the impact of the processes that we put foods and ingredients through, and some of them are inherent to the nature of the product. So it's really important to kind of understand that distinction about some of these are the structures that nature has designed, and some are the structures that come at the end of processes that we put foods and beverages through. So just to talk through some examples of natural, engineered, or processed structures, we've got fruits and vegetables, a great example of natural structures that consumers take and understand and consume often in the very form and structure that comes from nature. So here, a cucumber, often eaten raw, therefore, the structure that a consumer is experiencing there is entirely natural, no engineering or processing involved. Milk is an example of a natural structure, but also an example of an engineered or processed structure. So emulsions can be natural, obviously like milk, but also can be engineered and processed. Things like mayonnaise is also an emulsion, but it's an engineered emulsion. So mayonnaise doesn't appear in nature. Milk also often, certainly in the UK, is a processed structure. So most milk that you buy from the supermarkets goes through a process called homogenization. So that is a process that changes the structure of the milk. And the idea of homogenization of milk is you're making the fat droplets in milk, which is what makes it an emulsion, a much more even size. And that means that you don't get disproportionation of the fat droplets, and you don't see a layer of cream formed on the top of milk anymore. So those of you old enough to remember when we used to get milk delivered in glass bottles in the UK, often if you had full fat milk, you'd have a layer of cream at the top where the fat has risen to the top because the emulsion has begun to break down. Homogenization prevents that, which certainly in my eyes is a bad thing, because I used to love as a child that first part of the full fat milk where you had the cream on your cereal. And then we've got bread, which is entirely an engineered or processed structure, very different to what the ingredients are like in their natural state, and those ingredients go through a number of processes to create this processed structure of food, which is very much focused on the consumer experience. So things like texture, flavor, mouthfeel, all impacted by that engineered or processed structure. And all of these are going to have an impact on the availability of both macro and micronutrients. So we've talked about natural structures, we've talked about engineered, artificial, processed structures. What are kind of the main differences? So when we're looking at kind of natural structures, typically they are formed from things like protein, fat, and indigestible polysaccharides, things like cellulose, lignin, hemicellulose. And often in engineered and processed structures, those materials are also used. Whether it's plant proteins used, animal fats or natural fats, and fibers becoming more and more popular in processed products as the health benefits of fiber become clear. One of the big differences between engineered and processed structures and natural structures is the use of things like sugars and starches to create structures. Much less prevalent in nature, the use of starches and sugars to create structures, and that's because nature tends to be quite wet. Whether it's in living organisms or whether it's in the ground as things like seeds and grains grow, lots of water, and obviously sugars and starches tend to break down or dissolve into water quite easily, and that's not particularly useful to nature because it means that cells and structures will lose their shape, and the biological activities that happen in them won't necessarily be able to. So we like starch and sugar as humans. They're very tasty. A lot of the most popular products on the market have high levels of both of those things, but they tend to be processed, engineered in order to give the structures that we eat, and that has a significant impact on the availability of nutrients. And one thing to say, excuse me, is processing often disrupts natural structures. And over the next couple of slides, I'll take you through a kind of an example of how that is the case. So let's talk about an apple. Obviously, natural structure there on the left, an apple. And I've got a couple of different processed structures that you can create from an apple. We've got apple juice, and then we've got freeze-dried apple. These are two different structures that are created from a natural structure, and they both have significant impacts on the availability of the nutrients contained within the apple. So let's look at some of the nutrients that you find within an apple and the availability. So let's look at some of the bioactive compounds, so polyphenols and vitamins, both at quite a high level in apples. If you're juicing an apple, the availability of polyphenols and vitamins goes up significantly. You've broken down those natural structures that contain the polyphenols, and other bioactive compounds in the vitamins, and your body can absorb them really quickly, which is great. If you freeze dry an apple, it also increases the bioavailability, but you still have some of that natural structure there, so the availability is a little bit lower. Still higher than it is in a natural structure, because you've disrupted some of that cellular structure during the freeze drying, but not quite as available as they are in juices. One of the negative consequences, and this is certainly true for fruit, is the increase in the availability of glycemic carbohydrates, whether that's sugar in the case of apple, or things like starches in the case of grains. Your body is able to access the starches and sugars much more rapidly, and that causes a much higher spike in blood sugar in a juice than it does in a natural apple structure. And again, in freeze drying, it is not quite as quick, but it's still your glycemic index or blood sugar level will increase much more rapidly with a freeze-dried apple slice than it would with eating an apple. Asterisk next to vitamin availability, because processing often has an effect on the level of the vitamin, not just the availability. So it's always important to understand that the vitamins may be more available, but they also are more available to be oxidized or degraded by heat or acids that are contained within the product. So it's very much a double-edged sword. So, as I say, processing or structural modification is very much a double-edged sword. In the natural structure, you've sometimes got a higher level of nutrients because they've not been destroyed during the processing, and often a lower glycemic index. However, in the modified structure, you have things like shelf life benefits, you have consumer benefits, more appealing structure, the ability to use the product or ingredient in other formulations, often an increase in availability of some nutrients. So we talked about vitamins and other bioactive compounds, often more available if you disrupt that natural structure, but often comes with faster release of sugars and other glycemic carbohydrates, which is becoming more and more of a significant issue in health as the research continues. So, as you said, natural structures are good at holding on to nutrients. Some of them are positive, some of them are not. Natural structure is also very good at preventing the degradation of sensitive nutrients. But unfortunately, most processes disrupt those natural structures, but that's not always a bad thing. Processes can also be used to remove barriers that lead to positive health outcomes. So let's talk about some of the processes in the food and drink industry. So we've got milling. I know Paul's going to talk about milling in his presentation, so I'm not going to go into it into a huge level of detail. But you mill foods. We've been doing it for thousands of years to disrupt that cellulosic and lignin outer portion of the grains to access the digestible components within. So you're releasing those digestible carbohydrates by disrupting that indigestible outer structure. And potentially also, if you fractionate during milling, removing anti-nutritional components. Cooking and canning, you're disrupting natural structures by cooking, making potentially vitamins and minerals more accessible, as well as things like proteins and sugars. A great example of is lycopene in tomatoes is more available in canned tomatoes than it is in fresh tomatoes, and lycopene has been linked with a number of positive health benefits. And then we've got things like mincing. Makes protein more rapidly digestible in meat, potentially may increase iron and vitamin availability. Not a lot of data that I could find about that. But again, you've got these real benefits of processing natural structures in their ability to release more and more nutrients and also remove potential anti-nutrients. So what does that mean for a product developer? Well, one of the big questions we get asked at Camden BRI is, I've got two products, maybe an apple and a juiced apple, and I send them off for nutrition analysis, and if water level is taken into account, I get the same level of nutrients in both of these things. What does that mean for availability? How does that work? Well, there's quite a big difference between what the body does and what a nutritional testing lab does. In a body, you've got mastication, so you use teeth to break it down, and then as the product travels through your digestive system, it's attacked by a variety of different mechanisms that the body produces, whether that's enzymes in the mouth, acids in the stomach, And then finally, in the large intestine, you've got bacterial fermentation. Structure has an impact on that because nature has designed a lot of these structures to stop enzymes, whether they're pathogenic fungi or bacteria, attacking them. So there's some pretty good defenses there. In the lab, structure has little impact. We use food processors. We use, this is a SOX tech for looking at fat, which uses solvents to extract fat from products. It doesn't really, gives you a great idea of what's in there, but doesn't tell you anything about the availability because the human body is not a food processor. It doesn't use petrochemical solvents to extract fat. So the lab is very good at extracting every last microgram of a nutrient from foods, the body less so. So what does that mean and what's the significance? Altering the structure has many effects on the availability of nutrients to the body. Some of them positive, some of them negative. It very much depends product by product. Often the processing increases the availability of glycemic carbohydrates, which is one of the main downsides, but that isn't always the case. There are some circumstances where some processes actually reduce the availability of glycemic carbohydrates. And due to how samples are prepared and tested in the lab, back of pack nutritional information often does not truly reflect the nutrients consumers will benefit from. And that is true in micronutrients, and it's also true in macronutrients. Often it involves expensive testing with cell lines or animals or even humans in order to get a truly accurate understanding of availability. And more attention is being paid to the fact that what it says on the back of pack, certainly when with vitamins and minerals, often bears little resemblance to what consumers actually benefit from. Okay, so that was very much a whistle-stop tour. Say there'd be chance for questions and answers at the end. And I'm now going to hand over to Professor Paul Sharp from King's College London, who's going to talk about some of the work he's been doing looking at the bioavailability of minerals from wheat. And look forward to some of your questions at the end. Thank you. Over to you, Paul.
Thank you very much, Mike. I'm just going to share my screen. So hopefully you can all see that now. So as Mike said, I'm going to talk about micronutrient bioavailability. In particular, I'm going to talk about minerals and focus on iron and zinc and their bioavailability from cereals. And what I'm hoping to show you is that the cereals and cereal products that we consume in the UK provide a huge amount of the daily intake of iron and zinc. But actually the bioavailability of iron and zinc from those natural cereal products is quite low. So processing those products is really important if we want to maximize the bioavailable mineral from those cereals. So if I start by showing you how important iron and zinc, the contribution of cereals is to iron and zinc intake in different population groups in the UK. And this is some data from the National Diet and Nutrition Survey. And I've highlighted in particular the adolescent population group. And you can see that cereals and cereal products provide about half of the iron that's taken in on a daily basis by adolescent people, and about 30% of the zinc content. Now, if we try and look at that and see how it relates to the dietary reference values, what I'm showing you here is the reference nutrient intake, which is the upper level of requirements that's predicted, and the lower reference nutrient intake, which is the lower level. And in blue, the percentage of the population groups within the adolescent population whose intakes of iron and zinc are below the lower reference nutrient intake. And you can see for males, for iron, there's about 12% whose intakes are below the lower reference nutrient intake. And for zinc, about 18% of the male adolescent population in the UK have zinc intakes below the lower reference nutrient intake. And this is even more dramatic for adolescent females. For iron, about 50% of the population have iron intakes below the LRNI, and about 27% have zinc intakes below the lower reference nutrient intake. And what this means in practical terms is if your habitual intake of these nutrients is below the lower reference nutrient intake, then you're very unlikely to be meeting your metabolic requirements for these particular nutrients. So I guess the take-home message from this is that adolescents don't have enough iron and zinc in their diet to meet their requirements. If we try and relate this to mineral status in the UK and in the adolescent population group, then it's estimated that up to 30% of adolescent females in the UK are iron deficient, and up to 10% to 15% of the adolescent female population in the UK may also have zinc deficiency. So this leads us to a conclusion that if we could increase the bioavailability of iron and zinc from cereals and cereal products, then this might go some way to improving the mineral status in the adolescent population in the UK. So what are the barriers to bioavailability? Well, this is a slide showing a section through the wheat caryopsis, and for the data I'm going to show you today, I'm going to focus on wheat and wheat-based foods. And some of the factors that really limit the bioavailability of iron and zinc from cereals are the localization of the minerals within the whole grain cereal, the chemical speciation of those minerals in the plant, so what form is the iron and zinc stored in, and also importantly, the plant cell walls and how they can encapsulate the nutrients that we're interested in. And if we don't process those cell walls, then really we can't release the nutrients that we're interested in and digest and absorb those nutrients in the human gut. So this is a study that we did in collaboration with Peter Shurey and his colleagues at Rothamsted Research and the London Metallomics facility at King's College London. And this is showing the localization of iron and zinc within the wheat grain. And what you can see from the staining in these slides is that iron is largely present in the aileron layer and in the scutellum of the wheat. Zinc also highly prevalent in the aileron layer, but also there are very high levels of zinc in the embryo of the wheat grain. And then we've also got an overlay with the phosphorus signal, which you can see is the 31P panel there. And you can see that the phosphorus signal co-localizes with iron in particular, and also with zinc in the aileron and the scutellum. So the phosphorus is very prevalent in those particular structures within the wheat. We know that the phosphorus is largely present as phytic acid or phytate. And we also know that iron and zinc are largely stored within the aileron tissue as phytate complexes. And this presents a problem for human nutrition because we also know that phytate is the main dietary inhibitor of iron and zinc. So it will limit the amount of iron and zinc that we can absorb from cereal-based foods. So a combination of those factors, the localization, the co-storage in the form of phytate really limits the bioavailability of iron and zinc from cereals and cereal products. The other factor that I mentioned in terms of limiting bioavailability is the plant cell walls. And what we know is that plant cell walls are very resistant to digestion in the human gut. So I'm showing you two studies here that were carried out at King's. The first one on the left-hand side is from Peter Ellis and his colleagues, who fed a coarse wheat porridge to some healthy human volunteers who had an indwelling ileostomy, which is a tube at the end of the small intestine. And this allowed them to collect the effluent at the end of the small intestine. So the food had passed through the small intestine, through the normal digestive part of the gut. And what you can see in this image, when they collected the effluent at the end of the small intestine, there are still very large particles of starchy endosperm, aileron, testa, and pericarp present in that effluent. So those cell walls simply hadn't been broken down, and the nutrients that are contained within those cells would not have been released and would not be available for absorption. On the right-hand side, we have a study that we carried out using some wheat flour, and we fed this to mice. And you can see in the upper panel here, the wheat tissue, the typical wheat particles that we fed. You can see there's some endosperm there. And hopefully you can see that attached to that are some blue staining cells. So this is the alerone tissues, which are highly enriched in minerals, particularly iron. And this is what we call a pearl stain that is specifically detecting iron in those cellular tissues. And when we feed this wheat flour to mice, we can collect their feces. And you can see hopefully at the bottom where I've circled that some of those blue staining alerone cells are still present in the mouse feces. So this tissue has passed completely through the mouse gut, is excreted in feces. We still have intact cellular structure, and therefore any of the mineral that's contained within the alerone of this wheat flour will not have been released and will not have been taken up within the gut for future onward transport and metabolism in the body. So a combination of the speciation, the localization, and the encapsulation by plant cell walls really limits the bioavailability of minerals from cereals and cereal products. So what can we do to try and increase mineral bioavailability from cereal-based foods? Well, I'm going to show you some data from a project that was funded by the BBSRC through the drink funding scheme. And this also gives me an opportunity to acknowledge our collaborators on this project, who were Beulah, who provided us with some of the flour for the studies that I'm going to show you, Camden BRI, who carried out some structural work as part of this project, and also ADM Milling, who provided us with some white flour for some of the studies that I'm going to show you today. Oh, gone the wrong way. Okay, so the aim of the project was to see whether we could disrupt the plant cell walls in some way, and whether this would increase the bioavailability of iron from wheat-based foods. So Beulah provided us with 2 batches of flour, one which had been through a standard milling process, one which had been through a micronization process, and this is a modified roller milling process. And in the table, what you can see is, regardless of the processing technique, whether we've gone through standard milling or micro milling, then we're not losing any nutrients. So we can see that the iron, zinc, and phytic acid content of those 2 flours is pretty comparable. We're not losing any nutrient as a result of the differential processing methods. What we can do from there is we can bake bread. And for the studies I'm going to show you, we produce breads either containing yeast or without yeast. And we can subject those bread samples to an in vitro digestion process. So this is a fairly simplified model in which we incubate the bread samples with pepsin at pH 2 to simulate the conditions in the stomach. And then we raise the pH and add a cocktail of pancreatic enzymes and bile to simulate digestion in the small intestine. At the end of that process, we can spin down the food sample, we can collect the supernatant, and we can see how much iron and zinc has been released during the digestion process. We can also add some of that digest to a human intestinal cell line, and we can measure how much iron and zinc is taken up from those food digest by these cells. And this is our readout of bioavailability, how much of the mineral has been taken up by this cell line. And we measure that using ICP mass spectrometry. So here's some of the data. So we're looking now at the iron bioavailability, how much iron has been taken up by the human intestinal cell line, the KCO2 cells. And what you can see straight away is that in the breads that were produced with yeast, then there's more bioavailable iron released from those breads compared to the breads that were produced without yeast. And if we look at the within yeast group, so the breads that were produced with the yeast, you can see that the iron bioavailability from the micromilled flour breads is greater than that from the standard milled flour breads. So this is an indication that breaking down those wheat cell walls increases the amount of bioavailable iron that's released that can then be absorbed and utilized by the body. We also measured the phytic acid content of these breads, and I'm showing you in the slide, the figure on the right hand side, the original phytic acid level that was present in the whole flour. You can see that that is reduced even in the breads that were produced without yeast. And we think that this is because during the bread making process, the process of heating the dough and resting the dough, that activates the endogenous wheat phytase enzymes, and that will break down some of the phytic acid. And when we add yeast during the bread making process, yeast also has further phytase activity. So you get a further decrease in phytic acid content. So we think a combination of reducing the phytic acid content and breaking open those plant cell walls, they contribute together to increasing the bioavailability of iron from the micromilled flour produced breads. What about the other area then? So that was for a whole wheat flour. What about the issue of food fortification? Well, we know at the moment there are the bread and flour regulations in place, which require the mandatory fortification of white bread flour. And the mandatory level is 1.65 milligrams of iron per 100 grams of flour. And we also know that there's a voluntary fortification process where you can add iron to breakfast cereals, and that can be anywhere between 7 and 12 milligrams of iron per 100 grams of cereal. In the UK, we tend to use elemental iron powder for flour fortification. And the reason for this is that it has very few organoleptic properties. So you can't taste the iron in the food product. It doesn't discolor the product. It doesn't damage the shelf life of the product by causing any changes in oxidation of the flour. But obviously other compounds are available. So we could use ferrous sulfate or ferrous gluconate. These are very bioavailable compounds, but they have very high organoleptic properties. They'll cause discoloration of foods. They'll cause oxidation and reduce shelf life. And they'll also give you a metallic taste to the food product. So, okay for some highly flavored foods such as soy sauce or fish sauce, but not so good for a white bread flour. So we tend to use the elemental iron powder in preference. And you can see that this has got relatively low bioavailability. And that's largely because it's poorly soluble. So taking you back to a slide I showed you earlier, I showed you that the aileron is an important part of the wheat caryopsis. The aileron is actually botanically part of the endosperm, but it attaches particularly strongly to the outer bran layers, to the testa and pericarp, and it's removed during the milling process to produce white flour. The aileron contains something like 75 to 80% of the iron in the wheat grain. And the question we've asked is, could we add that aileron back into white flour to add as a natural fortificant? And this is, again, some in vitro work that we've done. And what we've compared is the aileron added into white flour versus elemental iron. And they've been added to give the same amount of iron in the flour. And what you can see is on the left-hand side, the bioaccessibility, so the amount of iron released, and the bioavailability of the iron from the aileron flour is greater than the white flour that's been fortified with elemental iron. So just to conclude everything then, I've shown you that cereals are major sources of minerals and also vitamins in the UK. Mineral bioaccessibility and bioavailability is limited from whole grain cereals, and that's a function of the localization within the plant tissue, and also the speciation of the mineral, the form in which it's stored in the plant tissue, particularly if those minerals are stored as complexes with phytic acid, and also the physical encapsulation by the plant cell walls, which are highly resistant to digestion in the gut, further limits the bioaccessibility and bioavailability of minerals. The bioavailability of the endogenous minerals can be increased by disrupting plant cell walls and also by reducing the phytic acid content. And we've got some evidence now from preliminary studies that adding back the endogenous mineral in the form of aleurone, adding that as a fortificant to white flour, may increase or may provide a bioavailable source of iron for food fortification. And with that, I'd like to thank you for your attention, and I'm now going to pass over to Eduardo for his presentation. Thank you.
Thank you, Paul, for giving the floor to me. First of all, thank you very much for inviting me today, and I'm very glad and proud to share with you some of the results I got in the last years of work in this field. So my research interest is in understanding the relationship between what we eat and the effect it has on our health. And of course, I did it along the years from, let's say, a food science perspective. And basically, the idea that was underlying my my work is that we need a change in paradigm here. The old paradigm in nutrition science, so when the relationship between what we eat and the effect on health is investigated, is that the health effect of our diets only depends on the type of nutrients or bioactive compounds that are there and their concentration. This is obviously, this is true, but it's not a whole picture, because in fact, we do not eat nutrients or bioactive compounds, but we eat food, and these foods are not just a carrier for these nutrients in our body. In fact, they are included in a food matrix, and when this matrix interacts with our gastrointestinal tract, it will modulate the health effect of our diet. And it does so by modulating the amount of nutrients that are available to our body, what we refer to in terms of bioavailability or digestibility, but also modulating the rate at which these nutrients or bioactive compounds accumulate in the lumen of the gastrointestinal tract or in our blood, which is also very important, because it can trigger different physiological responses and produce different physiological outcomes. So the paradigm shift that we need is that we have to include this food matrix in the picture when we want to understand the effect of our diets on our health. And when I talk about food matrix, what well, there are several excellent definitions of food matrix available in the scientific literature. This is the definition I gave a few years ago in one of my papers. So it's the chemical composition of food and the way the food components are structurally organized. So the food components interact with each other, producing structures at different length scales, so at a micro, meso, and microscopic scales. And my presentation today is very much focused on plant foods. And basically, when we have a look at the plant matrix, the most actually striking and important feature from a food matrix point of view is the presence of this continuous network of cell walls. And as been already touched upon in previous presentation, the cell walls are made up of carbohydrates that are not digestible by humans, so they cannot be digested by our digestive enzymes. And this means that they basically survive the transit through the gastrointestinal tract. And which also means that when the cell integrity is preserved at the point of swallowing, these cell walls can really act as a natural bioencapsulant and prevent the intracellular nutrients to be completely digested, as you can see in this cartoon here. So when the cell's integrity is retained, like in this case, the enzymes, the digestive enzymes, will have harder times to access intracellular substrates. Whereas when the cells are completely broken, the substrate is immediately and freely accessible to the digestive enzymes. So I will show you now a few examples of works that I have done or published in the scientific literature to see what will be the effect of this barrier bioencapsulation effect of the plant cell walls. Now, the first example is from a study that we performed last year. We were interested here in understanding, well, the effect of oral processing, so how long you chew on postprandial glucose peak, you realize that this is a very important indicator, because we know that the elevated continuous and elevated postprandial glucose peak may be associated with incidence of chronic diseases like diabetes. And in this study, we performed an acute intervention where we gave two different starchy food containing the same amount of starch to the subject. So we basically used brown rice and chickpeas. And after that, we took blood samples and we measured the level of glucose in the blood. And what we see in this picture, well, I will not talk about effect on oral processing. We might perhaps touch upon this later. But actually, the main differences that I want you to appreciate is the difference in the area under the curve of the postprandial glucose profile of brown rice compared to chickpeas, being the postprandial glucose profile of chickpeas much smaller. And the reason for that can be easily understood if you can have a look at the two foods at the microstructural level. So when you see the microstructures of cooked chickpeas, you see that most of the cell walls are still intact, which means that the amylase, which is the enzyme, of course, that is digesting starch, is impaired in reaching the starch, which is located within the cells. When you look at the rice, what you see is that after the cooking rice, all the cells are broken. This is because starch during rice contains a great amount of starch. So when you cook your rice, the starch will gelatinize, and the pressure will basically break all the thin cell walls of rice. So after cooking, you don't have any intact cells in rice, which means that the amylase can freely access starch, and the digestion of starch will be faster. Does this effect only pertain to starch digestion or carbohydrate digestion? Of course not. It also applies to digestion of other intracellular components, like for instance proteins. This is a study that we did a few years ago. We worked at in vitro, so we we performed an in vitro digestion in a very similar way as has been described by Paul in the previous slides. So we worked with soybeans, we isolated soybean proteins, and we studied the digestibility of soybean proteins in intact cells or in cells that have been mechanically broken. And in these graphs, you see the protein digestibility in time expressed as DH percentage, which is roughly the percentage of peptidic bonds that have been cleaved during digestion. And you see once again that when you break the cells, the protein digestibility occurs at a higher extent and much faster. And these in vitro data confirm data that are very present in the scientific literature since years. As you see in this table, you have here presented the true protein digestibility of 3 different forms of soybean matrix. You see that when you provide soybeans in the form of a protein isolate, like here, so no cell walls anymore, no barrier effect of the cell walls, the protein digestibility is very high, pretty much similar to protein digestibility of animal proteins. But when you increase the level of tissue integrity, moving from protein isolates towards flour or even intact soybeans, you see that the true protein digestibility will decrease. And the reason for that is that you have an increasingly higher fraction of intact cells, so this barrier effect is increasingly bigger. And whether you have a reduction of around 20% of protein digestibility, which is quite important from your body point of view, if you look at this difference from the perspective of what enters the large intestine, because you always have to consider that whatever is not digested in the small intestine will flow into the colon and will come into contact with the gut microbiota. It means that when you ingest the soybeans, you have 4 or 5 more proteins flowing in the colon compared to, for instance, soybean protein isolates. And the consequences on gut microbiota are still unknown because gut microbiota is known to ferment proteins producing harmful components. And of course, I mean, I also have an example about lipids, and this is a very early and a quite striking, outstanding example that you find is about almonds, and almonds are known to be quite caloric. If you see here the this table, the composition of a typical almond, you see that it's very rich in lipids. So when you apply the Atwater factors to calculate the energy content on almonds, as is in our regulations, you see that it's a very high caloric type of food. But actually, when the actual caloric content of almonds have been tested in vivo, these researchers already 10 years ago, they found out that the actual metabolizable energy from almond is more than 30% lower. So basically, the almonds are not as caloric as it would seem from the calculation of the energy content based on the back of pack type of information. And the reason for that, again, is that when you chew on almonds, you produce particles, and these particles contains a large fractions of intact cells, which protect lipids from digestion. This micrograph, you see a typical particle, and here you see stain in blue. You see all the lipids that are trapped within the particles that have not been digested. This is an image of the particle of almonds after in vitro digestion. So you see that most of the lipids are not digested because protected, locked inside the cell walls. So this also shows that at level of single items, single foods, the actual caloric content of these foods might not correspond to the one that we calculate and report in our back of pack. And a few years ago, we were actually wondering whether these would also apply to mixed diets. And so we performed this study 5 years ago. It was a desk study. We designed 3 different diets. We call it Western, Mediterranean, and vegan diet, including different and increasing level of plant food items or intact structures, meaning, for instance, cereals and legumes consumed as whole grains, fruit and vegetables, nuts. And what we did, we collected the data from scientific literature about the digestibility of carbohydrates, proteins, lipids in each item of these diets. We calculated the actual caloric content of these diets, and we compared them to the energy content as calculated by the regulation. So by using the Atwater factors for energy. And what we found was that you always have, when you include plant tissues, intact plant tissues in a diet, you always have an overestimation. And this overestimation is proportional to the amount of plant tissues, intact plant tissues that you included in the diet, meaning that the actual caloric content of these diets is always lower than what is theoretically calculated. And the reason for that, once again, is that part of this energy is lost in the feces, trapped within intact tissue particles and intact cells. And this basically brings me to my conclusions that are the integrity of the plant tissues that modulates the bioavailability of nutrients, all of them, carbohydrates, proteins, and lipids, and then also the actual caloric content on food and diets. But it can also modulate the kinetics of nutrients absorption. And I gave you the example of the postprandial glucose profile after feeding rice and chickpeas, for instance. And as obvious consequence, I mean, process technologies like milling and grinding, which increase the fractions of broken cells compared to intact cells, also increase the amount of nutrients available for our body, and from the perspective of the large intestine, reduces the amount of these nutrients available for our gut microbiota. And this is my end of my presentation.
Thank you so much, Eduardo. So we're going to move to the Q&A part of the session. So if I can invite Paul and Mike to also come back on camera, that would be great. We do have some live questions and actually not too much time, so I will try and rattle through a few. But Mike, if I could come to you first, because I was actually going to ask you about the way of, you talked about the natural structures and actually whether examples of more kind of engineered structured, which would maybe help reduce that GI response that you were seeing. And actually somebody in the Q&A had also asked about how is that kind of knowledge of structures that is obviously building being put into R&D work at the moment?
That's a great question, Kate, and a great question. I don't see who asked it, but it's a great question from the audience. The key aspect of that is not so much the structure as what the structure is made of. So as I talked about, a lot of food products are, the structure is made from things like starches and sugars, which are rapidly digestible by the human body. We're seeing more and more interest and more companies looking towards using things like fibers in their structure, which are either indigestible or much more slowly digestible by the body. And it's about, you know, trying to match the existing structure, because often it's an existing food product that consumers know and love, and the company business makes a lot of money from. And it's about matching those structures using other compounds that slow down the release of kind of the unwanted nutrients. You know, we've talked a lot about, you know, altering natural structures will increase beneficial nutrients. A lot of the work that I deal with is focused more on the macronutrient kind of, and a lot of the work that Eduardo mentioned in his presentation. You know, the industry is really catching on to the fact that what's in the product isn't as important as what the body takes from it. And we're seeing more and more companies looking at how they can slow down the certainly things like the glycemic spike from foods. Part of that structure, but more importantly is what the structure is made of, and how we can use these new and emerging ingredients to deliver the same kind of benefits to the consumer, so taste, flavor, texture, whilst making them healthier without, you know, and trying to get exactly the same product, which is a big challenge.
It's quite a challenge.
Yeah, quite a challenge.
I'm seeing quite a balance actually between, as you say, there's that kind of, you know, that real balance of micronutrient and macronutrient, and what are the nutrients you actually want to have greater access to, as well. Paul, Alarone seems to have created quite a lot of interest in the Q&A, so I'm going to bundle a few up together for you. So there was a question around the impact on zinc bioavailability, the impact on the taste, and what the cost implication is compared to fundamental iron fortification.
Yeah, sure. So the effects that we see seem to be most prevalent for iron. When we look at zinc bioavailability measures, there's really not that much difference compared. There obviously is, because you're adding extra zinc in, but you know, in terms of enhancing the bioavailability, it doesn't seem to be that much great. And I suppose there may be some added benefit because we don't currently fortify foods with zinc in the UK, you know, so we would be adding in extra zinc. And I guess that would be true for a lot of other vitamins and minerals as well. You know, the aileron's not just rich in iron and zinc, it's also rich in calcium. It's rich in a whole range of the B vitamins that we currently add to white flour through fortification. So there could be a much wider benefit in terms of nutrition by providing a whole sort of cocktail of important vitamins and minerals into white flour. In terms of the taste, we haven't done any taste tests with it, but I mean, obviously one of the things to bear in mind is that the aileron is particularly rich in lipids. So potentially that could have an impact on shelf life. It could have an impact on the oxidation of those lipids, which would have a knock-on effect on flavor as well. And really those sort of sensory studies would need to be done before you could take this work any further. And the cost implication, I guess, you know, there is that implication, because at the moment we're using a purified aileron flour. What we haven't looked at this stage is whether, for example, you could just add a coarse bran back in there, and whether that would have the same effect, because you'd be adding the aileron back in with the bran, whether it would have the same sort of beneficial effects in terms of iron bioavailability. But yeah, very start of the journey, as they say. You know, there's a long way to go before we could think about product development.
Yeah, absolutely. And then kind of zooming out a bit, I think this might be certainly in the first instance to Eduardo, but others may want to come in, because this is talking about, there's a question effectively about bioavailability, but taking that broader picture, so not just looking at an individual ingredient, but the question is really about how complicated is it? So you zoomed us out to diets. So they do ask about what the nutrients consumed with, but also, do we have any idea about things like gender and ethnicity and time of day and kind of all those other aspects around the bioavailability of the food?
Yeah, definitely. First of all, I would like to stress to all the ones that are following the presentation, if they have, we don't have time to answer all the questions. If they really, they can write to me an email. I will be very glad to answer all their questions, because I see many actually appearing in the Q&A. Yes, there are inter-individual differences in the bioavailability related, for instance, on the way we chew our food. Depending on the level of bile acids that are produced and digestive secretions, the transit time. Of course, I mean, this is more like type of nutrition or physiologic type of work. I'm more focused on the variability in bioavailability that the food science can produce. So how we can modulate or we can manipulate our food matrix to increase or decrease, because it depends on also on the type of nutrients. Bioavailability, digestibility, and also the amount of nutrients that are flowing into the large intestine where the gut microbiota is actually living. So yeah, definitely inter-individual differences, but well, it's still a relatively virgin area, I would say.
Yeah, absolutely. And then there is a question which I think is to Paul, because there is a question about the comparability of rodent metabolism to humans. So I'm guessing that's referring to some of the earlier studies you were showing.
Yeah, so I mean, I guess the sort of structure of the mammalian gut is fairly similar throughout, you know, the different organisms. You know, that there are some subtle differences in there, but you know, that was really just to show that transit through the gut, you know, even going right through the colon where you've got the microbiota present, there's still a significant amount of the tissue remains intact, even through in the mouse feces. I'm not aware that anybody's looked at that in human directly, but certainly from the work that Peter Ellis and his colleagues have done, there is definitely, you know, digestion doesn't seem to, the cell walls don't appear to be digested during the transit through the small intestine. And of course, for iron absorption, it's really important that the cell walls are broken down early, because virtually all of the iron that we absorb right in the first part of the duodenum. So as the food is literally leaving the stomach and entering the small intestine, the highest concentration of the transporters that are involved in the absorption of iron are in that very proximal part of the duodenum. So if you've got to the end of the small intestine without breaking down the cell walls, then in terms of absorbing iron, it really would have no impact if they were broken down in the colon, because we don't absorb iron in the colon to any significant extent.
Right, okay, thank you. And I think I'm going to ask one last question and take Chair's prerogative to make us overrun by about 3 minutes. Understand if people have to drop off before that. But there is a question about nutrition information on pack. Which the question's actually directed to Mike, because I guess because you started, Mike, but around that, you know, the information on pack we're giving might not actually be correct effectively in terms of what we're digesting, but maybe both Mike and Eduardo, you might want to comment on that.
Yeah, it's really difficult, because we need to have simple, quick, cheap tests that food companies can use, but it is, you know, it is how we prepare samples in lab and how we test them is nowhere near how the human body breaks them down and absorbs them. I mean, a great example I heard was from Professor Giles Yeo at Cambridge, I think, or Oxford. I'm sure I've insulted him by getting that wrong. He talked about a raw stick of celery has 6 calories. That same stick of celery, if cooked, has 30 calories. Now, if you tested it in the lab, the likelihood is it would have 30 calories, because, you know, that a lot of that celery will be passing through your body without you accessing the nutrients if it was raw, whereas cooking allows you to access them. The lab will effectively break down a lot of the structure and will gain all of it. So, you know, it is a simple fact that most back of pack nutrients, whether it's macro or micro, doesn't really give you an indication of how much of any of those your own body will get. But it varies so much between, as we've talked about already, individuals, and also what time of day, what you're eating it with, what you ate before, that it is the best we can do without having to test each combination, which is obviously infinite, with each different body, which again is infinite. So it's the least, it's an imperfect system, but it's probably, as a comparative, it gives enough information that you can make a broadly informed choice.
Can I add something to this nice word? Yeah, I think I totally agree, and definitely the system we have to calculate the energy content of food is inaccurate. But what I was surprised is when you approach the thing from a mixed diet point of view, if the diet is well balanced, I mean, the differences are not that big. As I showed in my, I mean, we really prepared a very extreme diet, the vegan diet, a lot of plant intact structures there. And the difference was just 7%. So it's not negligible. I mean, 200 calories per day in a vegan diet might be something. But when you have a balanced type of diet, then this discrepancy might not be huge. And perhaps a way to go is in the US, they use specific water factors that are specific for each food and each energy yielding substrate. I think if we can combine that approach to our way in Europe to calculate the energy content, we might get closer to the actual caloric content of food.
Yeah, interesting. And I did think when I saw that slide of yours, the kind of course, for research purposes, is you need almost extreme examples, and then, but actually, if we look at what people eat, it will make even less of a difference than that 7%.
And of course, there's no information about micronutrients on the back of pack either, and that's a whole new can of worms, because it also doesn't just depend on content, it also depends on what else you're eating as part of a meal that will influence the bioavailability massively.
Yeah, and coming back to that, the structure in terms of the maybe giving us a beneficial impact in terms of, say, micronutrient content, but then an impact you're maybe not looking for in terms of glycemic response. So balancing all of that up together as well, which is why this is such an interesting topic. But unfortunately, I am going to have to draw us to a close. If we didn't manage to answer your questions, then all of the speakers have said that they are happy to be contacted directly. And you will see emails on the screen now, so you will be able to email people. We will be sharing a recording of the webinar, which includes the Q&A session, to everyone who has attended today, and it will also be available on the IFST Webinar Hub, which is ifst.org/webinars. There you go, you can see it on your screen now. Please, can I ask, before you log off, that you do fill in a very short survey, which will launch automatically. It really just helps IFST in looking at the types of webinars we should be offering and what they should be doing for you in the future. And all that it remains for me to say is to thank once again Mike, Paul and Eduardo for really interesting presentations and discussion afterwards, and thank everybody who has attended today.
Thank you very much, Kate.
Thanks, everyone. Thanks, Kate.
Day.